Hello students, here's a little video to give you a bit of support after you have completed the digestion and absorption pack. We're going to run through some of the trickier concepts and I'm going to show you a couple of activities you can do to consolidate it. And I'm going to show you a way of accessing support, getting some questions answered basically. So this is the PowerPoint that you have loaded up to go alongside your booklet. And hopefully you've been through the booklet, didn't pose you too many problems. Now I'm going to fly through the stuff here that I think is easy, and I'm going to focus on the stuff that is difficult. So the first bit is all about the structure of the digestive system, and it is self-teachable. You have to learn a bunch of words, true, but if you can do that, you can actually just internalise this information. This little diagram here, obviously you don't need to be able to draw it exactly line for line, but you need to know the order that things go in, okay? And you also need to be aware of the small intestine being divided into these sections, the duodenum and the ileum. So it was duodenum first, then ileum. It's in alphabetical order, if you like, and what happens in each of those. So basically, the duodenum is where the stomach contents goes after it leaves the stomach. It's also where the gallbladder is going to be able to deposit its bile salts and some bile, so you neutralise the acid. And the ileum is where a lot of the absorption of nutrients happens. Okay. So I'm going to skip past all of this. You can look it up and fill in anything you need. And even this stuff here about physical and chemical digestion is pretty easy. The digestion stuff is all a bit GCSE-like. It talks about enzymes and the process of breaking down large molecules, like starch, into small ones, like glucose, that we can absorb. What we do is we go into the detail of the stages. And most big molecules have this kind of stage here, sort of intermediate stage, where they're not fully broken down, but they almost are. So starch, for example, gets broken down to a disaccharide first, and then you use membrane-bound disaccharidases, and they're called maltase incidentally, because of course the disaccharide will be maltose to produce glucose at the end. This is all biological molecule stuff. Okay? This is all linking back to that first topic. If you didn't know that chopping starch up into units of 2 sugars would give you maltose, you need to go back over and learn your disaccharides. Okay, so we do that for the carbohydrates. We talk about the membrane-bound enzymes. We do the same for proteins, and the proteins are very similar. Look, you start with a big long chain. Here's your intermediate bit where you chop it up in the middle with your endopeptidases, and then your exopeptidases get to work removing dipeptides from the ends of those short fragments, and those dipeptides can then be hydrolyzed by membrane-bound dipeptidases, again in the ileum. Okay, so it's very similar to carbohydrates. Lipids, a bit weird, because different structure, they're not a polymer. So the lipases will be breaking down your lipids into fatty acids and glycerol. And weirdly, they don't always go all the way. Sometimes they stop at a monoglyceride phase. So they break off 2 of the fatty acid chains. More on lipids later on, but that's basically what we need to know there. Now the bit that people find difficult here, I've noticed, tends to be absorption. So we're going to go through some absorption stuff. Now the PowerPoint lays it all out there, and you need to know how you absorb proteins, carbohydrates, and lipids. Now obviously if you're absorbing proteins and carbohydrates, you're not absorbing like a whole protein or starch. You've broken them down to component monomers, but you need to know the process of getting them into your bloodstream. So this is where co-transport comes in. Now co-transport is a process that you need to know. This diagram here and this explanation, that is it. That's what you need to know, and it's really worth learning it now because, spoiler alert, it comes up again in the upper sixth. So I'm going to go through a little activity that you can do that actually might make it hopefully stick in your head, or something you can practice at home at least. Okay, so here's the old visualizer. The principle here, we're gonna sketch it out in really simple terms. Okay, so here I have I'm going to just label this as the lumen. This is basically the intestine. Yeah, this is where your food is coming in, and then we're going to have some big old ciliated epithelial cells here. Like that. So let's do 2. Although in truth, I only really need 1. Okay, and then here is a capillary. So what we've got to do is we've got to get any sugars from here into here so they can be taken away by the bloodstream. Let's have the blood flowing in that direction like that. Now I'm going to do this for monosaccharides. I'm going to do it for glucose, but it works exactly the same for amino acids. So this is one of those things. Learn it once, get to use it twice. Okay, you can do it for amino acids and glucose, and as I said, you need to know it now, but it also happens in the upper sixth of a different part of the body. So this is such a valuable thing to be able to learn and do from memory. Okay, I've put quite a high concentration of glucose up here. Right, we've just eaten a meal, so we've got lots and lots of sugar being or starch being digested. And the thing is, you couldn't just rely on good old facilitated diffusion. If you were to put in a channel protein here and have this diffusing until it reached equilibrium, you'd end up with 50% of the glucose being absorbed, 50% remaining in the intestine, and well, you'd end up with 50% of the nutrients you eat just passing straight through the body. That's not efficient, okay? That's not what we want. So what we've got to do is we've got to find a way of taking all of this glucose into here. And that means at some stage we're going to end up going against a concentration gradient. Right, that's our first thing. So in order to do this, we can't rely on passive processes. It's got to be active. And we're going to use this active process of co-transport. So it's not active transport in the pure sense. It's actually co-transport, which is a variant of active transport. Let's have a little look at how it's going to work. Right, let's put some proteins in the membrane. Now this one here is known as the sodium-potassium pump. It uses up ATP, okay, which means that it requires energy to drive it. And what it does is it pumps sodium ions out of the cell, and it pumps potassium ions into the cell. Let's add a dash of colour, shall we? Here are some nice green sodium ions. They've been pumped out of the cell into the bloodstream, which means they are whisked away. The potassium that we pump in here doesn't really play any future role in proceedings. So actually, from this point on, I'm going to ignore the potassium. Yeah, you're going to build up a concentration in here, but that's fine. Doesn't matter. The key point is, any sodium that was in here is going to be whisked out of the cell. So there is now a very low concentration of sodium in here. And any that ends up in the bloodstream is instantly removed. Why are we doing this? Because we're not after sodium, we want glucose. Well, by removing sodium, what happens is you set up a concentration gradient, because the food and drink we consume always contains sodium ions. It's a component of salt, sodium chloride. It's just on and in everything we do. So there's going to be absolutely tons of sodium ions up here. Now that we've established a concentration gradient, we can use it like a store of potential energy to drag glucose into this cell. And if we're going to do that, we need a special protein. This protein is known as a symport, because what it has are two binding sites. One binding site for a sodium ion, one binding site for a badly drawn glucose molecule. And when both are bound, it will change shape, drag them onto the other side of the phospholipid membrane, and it will release the glucose onto this side, and therefore in the cytoplasm. It doesn't use ATP. It's dragging glucose against a concentration gradient eventually, but it finds the energy to do that by using the potential energy of this sodium ion gradient. So what this is going to do is it's going to absolutely load this cell with glucose. All of these can be dragged that way. So you can have a very low concentration in the lumen, because you've almost absorbed it all, and a very high concentration in the cell. But crucially, as long as there is a sodium ion concentration, you will keep transporting glucose in this way. That means that once you get a very high concentration here, you can actually get it into the bloodstream through good old-fashioned facilitated diffusion. So you put channel protein in there, these guys can diffuse across the plasma membrane into the capillary, away they go around the blood circulatory system to your respiring cells doing whatever they want. Okay, and that is co-transport. Okay, so look, you've got a couple of things. You've got some active transport going on here, right, in this region. You've got this bit here, co-transport, so this is the sort of magic bit here, and then you've got a bit of facilitated diffusion going on here. So there's kind of three things happening. You do need ATP to make it work. The second you stop having any ATP, co-transport will stop, but it's a really good way of getting all of that glucose into your bloodstream. And, fun fact, it works exactly the same for amino acids. So I could have drawn another shape for amino acids, okay? So, and you'd have a different syn-porter, co-transporter protein here. Sodium ion will bind to that. The amino acid binds there, that drags it in there. You'll have another channel protein there. You also still have your sodium potassium pump there. So exactly the same thing works for amino acids. I've got this cell doing glucose, that cell doing amino acids. In actual fact, the cells of the ileum will be doing both. They're all involved in absorbing it. Okay, so that's the absorption of proteins and carbohydrates. Okay, hope that's helpful. Now, you need to practice drawing this. You need to get to the stage where you can do this from memory. Okay, so there's just one more bit to look at, and it's the bit that nobody likes. It's the bit where you have to talk about the absorption of lipids. Now, we know lipids are weird, right? Because they are insoluble. They form these droplets. So, lipids have absorption in a slightly different way. Now, they don't need to go through proteins. Because they're lipids, they will just dissolve perfectly happily in the phospholipid bilayer. But what you do need to do is ensure that you can break down the triglycerides effectively into monoglycerides and fatty acids. And the problem you've got here is that fat, when you eat it, it forms massive great globules or droplets in your digestive tract, which is where these bile salts come in. Adding bile salts emulsifies these big fat droplets into very small ones, and that gives a much larger surface area for your lipases, your enzymes, to get to work. Then you'll have your fatty acids and your monoglycerides that can be formed into these nice cells, and then they will just be able to diffuse across the plasma membrane. So far, so good. The tricky bit comes when you try to get them out and into the bloodstream, because you've got the same problem. They're lipids, they don't like dissolving in fluids. So actually your digestive system has evolved to come up with a sort of workaround here. What it does is it takes those monoglycerides and fatty acids and it processes them. And sometimes they can be reformed into triglycerides in the epithelial cell. And those triglycerides can be packaged up inside a little protein bundle called a chylomicron. Now, chylomicrons have proteins around the edge, which means that you can actually put them inside a vesicle. And a vesicle can then move to this membrane, the opposite membrane from the one in your intestinal lumen, fuse with it, and you will release that little protein bundle filled with lipids into this interstitial space here. So it's just a gap between two cells, at which point it will diffuse into this, which is called the lacteal. And the lacteal is part of your lymphatic system, which is a secondary network of tubes that runs all over the body. If you've ever had an infection and you get kind of big swellings, sort of in your jaw, like tonsillitis, you get your neck swelling up a little bit, that's part of the lymphatic system there, because it also is involved in the immune system to some extent. But the lacteal links with the lymphatic system, which is a network of tubes that travels all the way through the body. But crucially, what it's going to do here is it's going to take these chylomicrons from your digestive tract, and there's a point up here where your lymphatic system joins the circulatory system, the blood, the capillaries, arteries and veins. It's called a thoracic duct. It's kind of right whack bang in the middle of your body, just above the heart. And what you do is you steadily drip feed those triglycerides in there. So the reason you have to do all of this is because they are not water soluble. They're lipid soluble, so they form these droplets. Okay. So I think the key diagram for that one is this one here. If you had to learn one, learn this. Alright, I've blazed through a lot of information. Don't forget the links here. I'm going to post a couple of tasks that you can do that will involve a little bit more extended writing to explain and contrast the differences between these types of absorption. And I have created for you a web page where you can post your questions. So the link for that will be also sent to you guys. Basically, you can put post-it notes up on this page here. They will appear and I can write answers underneath. Now,